Okay, last day, finally, and tomorrow is going to be a good one, right? We're going to be ready for this part at least. I need to focus and stop getting sidetracked so much, but I will promise you we'll get through all the material today that you need for the exam tomorrow, okay? I've seen the exam questions, so I know exactly what I need to teach. That's perfect. The one thing that I thought I'd do is if there's time, and even if there's not time, I'll have 3 example exam questions at the end. How does that sound? Sounds good? So if you don't get to those in class, then I'll post them on Canvas, or I'm Canvas illiterate, so I'll have somebody, one of the TAs post it for me, okay? Okay, so instead of doing a whole recap of what we talked about yesterday, I think we kind of got it, right? We had male reproduction, we kind of went through all that stuff. Again, hormones, pretty straightforward. Anytime we talk about a hormone, and what would be a fair question on the exam is that, well, let's just practice one. Everybody's writing out, oh, here's an example question. So you already know this is not going to be on the exam then, right? Which of the following would have a receptor for AMH? Sertoli cells, Leydig cells, germ cells, an epithelial cell of the Wolffian duct, or an epithelium cell of the Mullerian duct? So A, B, C, D. I think that's it, or maybe I missed one. Which one? What do you think? A hint for multiple choice exams, at least for mine, a strategy is process of elimination. If you know it's wrong, it's wrong. Cross it out, move on, okay? So, let's do it. Sertoli cells, yes or no? Do they have a receptor for AMH? No? Who says no? Who says yes? Okay. No, why would they? Have we talked about what AMH does to Sertoli cells? The only thing we said was AMH secretes, or Sertoli cells secrete AMH. That's it, okay? What about Leydig cells? Do they have receptors for AMH? Raise your hand if you say yes. Raise your hand if you say no. Raise your hand if you say, I don't know. Okay, good. Again, have we talked about AMH doing something to Leydig cells? No, then they don't have a receptor for AMH. Okay. Germ cells, do they have a receptor for AMH? Exactly, no. Epithelial cell of the Wolffian duct, does that have AMH receptors? Yes? Why? What does AMH do to the Wolffian duct? Yeah, but I'm talking about the Wolffian duct. Are we getting it? There's no receptor for AMH on the Wolffian duct. The Mullerian duct responds to AMH, yes? It gets rid of the Mullerian duct. AMH causes the Mullerian duct to disappear. That means AMH, or receptors for AMH are on the Mullerian duct. Does that make sense? Okay, good. And then we started female reproductive physiology, okay? And so we went through meiosis, and again, I showed that one slide, and I forgot to post that. If you want, I can do it today. Do you want me to post that meiosis 1 and meiosis 2 versus mitosis doesn't matter? You guys kind of get it? I can post it if you want, okay? But kind of keep in mind, again, compare male and female germ cells. When does meiosis 1 start? When does it finish? When does meiosis 2 start, and when does it finish? In male germ cells, we constantly divide cells, so they get half of every material. In the oocyte, what does it do? Does it divide, or does it is there a different way that it gets rid of that chromosomal material following completion of meiosis 1 or 2? It does different, right? You have something called polar bodies. Basically a vesicle, a garbage bag that the oocyte is like taking out the trash. Okay, so you just get a first polar body, which will have all 23 chromosomes, but with the butterflies, right? They have twice the amount of DNA. And in the first polar body, we'd have the same thing. A second polar body after fertilization, because then we complete meiosis 2, will have the chromatids separate. So now we're also still 23. Okay, make sense? If not, then now I slowly see panic setting in. Oh crap, I need to know all this stuff for tomorrow. You'll be okay. Okay, so we're going to continue. We're going to get into a female reproductive cycle, okay? So the key thing is some things to keep in mind, and I don't know if you want to write this down. Hormones. Always ask yourself, where does it come from? Where does it act on? Okay, what cell type? And then obviously, what does it do? Okay? And the difference between female and male reproductive physiology is that in female reproductive physiology, it changes constantly. And if you kind of think about it, take a step back, big picture stuff, that makes sense, right? If you talk about the female reproductive cycle, what is basically the body of the woman doing every month? Well, a lot, but related to the reproductive cycle. What's the whole point of that? Sorry?
To release an egg.
Yeah, so ovulate an egg, but males do that too. Male release sperm all the time. We talked about that, so that's not really unique. What is basically the female body trying to prepare for every single month? Pregnancy. Every month, all the stuff that the woman's body is doing, the female reproductive tract, is getting ready for a potential pregnancy. Okay? Now, is a woman getting pregnant every month? No. Okay? So that means we have to try that over and over and over and over and over again. Okay? Because the great majority of time, there's never any sperm around to fertilize anything. And even if there is sperm around to fertilize something, a lot of times that never actually implants or develops. Okay? So it's pretty amazing what a female reproductive tract does every single month. Okay? I've heard it's a great nuisance and stuff like that, but trust me, it's a good thing. Okay? Okay, so really quickly, one of the things that I wanted to think of, we talked about the fact that 90-year-old males can still make sperm. And pretty much it stops when men are dead, right? Is that the case for females? Do they always make eggs and more eggs and more eggs, and there's plenty of eggs to go around? No. How do you know? Everybody said, no, there's not enough eggs. So how do you know? Well, it's not like you can look at side and see how many eggs there are. This figure will kind of tell you how many eggs there are, but how do you know that a woman runs out of eggs? Menopause, exactly. Menopause is a sign that a woman doesn't really run out of eggs. There's so few eggs left, maybe a couple of hundred, that it's not enough to start another cycle. And basically the ovary becomes quiescent. Okay, it's not going to respond anymore. There's no more ovulation, and the ovary is done. Okay, so contrary to some scientific papers that came out several years ago that got a lot of popular press and highlights and stuff like that, that there may be some kind of a stem cell-like thing in the ovary that could generate an egg, that is still very controversial. Even if that was the case, and a lot of these studies are done in mice, and mice are always weird because you can do pretty much anything you want with a mouse. Okay, even if that was the case, even if there was some very primordial progenitor cell in the ovary of a woman that could give rise to an egg, are women still undergoing menopause? Yes or no? Yes. Okay, so really practically, at some point, eggs are not activated anymore. We're done. Okay, so that has implications for many things. Okay. Childhood cancer in girls. Okay, usually that involves very harsh chemotherapy that in some cases, or many cases, can wipe out, destroy all the eggs. Is that a problem? Yes, because if you're 12 or 15 or 16 years old, you will never ovulate, you'll have ovarian failure, and that's it. Okay, there is no replenishing of stuff. Yes?
In those cases, like, does their body acknowledge that there's no need to ovulate? Or do they still go through, like, regular cycles?
No, there won't be any cycles. So basically, in those cases, women that have, girls that have premature ovarian failure, they are in menopause. Okay, that's pretty much it. Okay, so this graph kind of explains what's going on, and it's kind of, this is, the reason behind this is still kind of elusive to scientists of why, and this is really difficult to study in humans anyway, right? When did germ cells enter meiosis in the ovary, if we wanted to study that? When does that happen? They enter and arrest in meiosis during what? Fetal life. Can you study that? How would you study that? We're not studying fetuses, right? So this is kind of a timeline. Here we have about birth, puberty, and menopause. This is a little wrong. Obviously, it never is 0. But basically, initially, we talked about germ cells proliferating more and more and more, and up to a point that a woman can have up to 7 million eggs. Okay? And that is about 7 months of pregnancy. Then you start entering meiosis 1, and they arrest in meiosis 1. That takes a little bit of time, and that's it. Not dividing anymore, and that's it. And then what happens by the time of birth? What happens between the enter meiosis 1 and birth, according to that graph?
You lose a ton.
Yeah, you lose most of them. By the time a baby girl is born, she'll be down to 2 million. Why is this happening? Honestly, we had no idea. Okay, so initially, these eggs kind of are all stuck together. They kind of form nests. Okay, I didn't talk about that on Tuesday, and you can kind of ignore that. The idea here is, because this is really speculation, is then we will start forming these primordial follicles, right, that I talked about. This tiny little, not fully grown oocyte yet, and it had like a flat 1 or 2 granulosa cells around it. So basically that means that during that time period, when they enter meiotic arrest, they start forming these primordial follicles. And there's some thought that this process of breaking down these nests, okay, if that doesn't happen correctly, they just die off. Okay, but again, how do you study this? We cannot study babies that are 7 months along during pregnancy. Okay, then we go to puberty. We lose more. So by the time actually a woman is reproductively, I guess, active, you know, in terms of that she could ovulate and things like that, we already are down to maybe 500,000. And then throughout her reproductive lifespan, less and less and less. Okay, so every month, so if we start puberty with, that's the first cycle. Okay, every month, one thing that I want you to start kind of thinking about is, it is not, you saw all these different follicle types that we talked about yesterday, right? So understand which ones are which. We talked about an early antral, an antral, a mature, and all these things. Every month what happens is, about in women, is that about 10 to 25 follicles are picked, okay, that are present. So every month, 10 to 25. It's kind of between in that range. And that month, that group is going to grow and get bigger. Up to a point, till you're about midway through the monthly cycle, actually after a week, one gets selected to continue growing. Not 2, not 10, 1 out of that group that started growing that month. 1 is selected to continue grow. That's the one that's eventually going to ovulate. What happens to, let's say you have 12 that month. 1 continues to grow. That's the one that's going to ovulate midway through the reproductive cycle. What happens to the other 11? They die. Okay? So every month, you get 1 to grow and ovulate, which is great. You also have more eggs dying off. Okay? Does that kind of make sense? Okay, so you're going to keep going and going and going until menopause hits. When do women reach menopause? It's like puberty. Okay, so what, 45, 50, 55, 60? I have no idea. Plus, I'm not, the only thing I remember from menopause is I was already in the US and my parents live in the Netherlands, and my dad complaining on the phone like, your mom is sitting outside, it's snowing, she's so hot. I said, well, I think she has menopause. Okay, these hot flashes. Okay, anyway, I'm getting sidetracked already. Okay, let's do the hormonal regulation. Okay, so, I'm going to draw this out a little bit, kind of what we did yesterday. Here we go. We started with the 2 hormones. Again, everything starts with GnRH. Okay, so I'm just going to, yes, there's a question.
Sorry, before you launch into a new thing. Since birth control tricks the body into not ovulating, does it push back menopause?
Very good question. We're going to get back to that. So I like that question. That question shows up every single time, and we're going to talk about that. Okay. So starting with the anterior pituitary gland, LH, FSH. Okay. LH acts on theca cells. Okay. This is very parallel to male reproduction. LH acts on theca cells causing testosterone production. This is like LH acting on Leydig cells causing testosterone production. And think back, 2 days ago we talked about Leydig cells and theca cells coming from the same cell type. Okay, FSH acts on granulosa cells. Okay, and does a couple of things, just kind of like what FSH did with Sertoli cells. Okay, the first thing that happens is granulosa cells will secrete inhibin and causes negative feedback. Very similar to the male, except change granulosa cells to Sertoli cells. Okay, so really there's nothing new here, just words. Okay. Okay, the first thing that happens is testosterone from the theca cells, and again, look at that structure. The theca cells are all the way on the outside. Testosterone diffuses towards the granulosa cells because they're more on the inside. Okay, so testosterone goes to granulosa cells, and granulosa cells will take that testosterone and convert it into what? What do you think would be a good thing to make in ovaries? A hormone. Estrogen, there we go. Okay, and the way that works really is, so basically what I'm going to do now is E2, estrogen. Okay, remember that, did I mention that? I don't think I mentioned that enzyme involved with that. It doesn't really matter too much. It's called aromatase. Aromatase takes testosterone, converts it into estradiol. Okay, so, but you need FSH for that. Okay, FSH actually activates that enzyme. Okay, so initially we have these small little follicles. So this happens, right? So we have a little bit of estrogen production by all these granulosa cells. They're not that many yet, so there's a little bit of estrogen from growing follicles. So in a particular month, this could be 15 growing follicles. Okay, this little bit of estrogen has a negative feedback to the brain. Okay, negative feedback to the brain. Now, out of these growing population of follicles, I mentioned one is selected. Okay, that continues to grow, the other ones die off. And that continues to grow and grow and grow. So what happens to estrogen levels? They go up and up and up. Do you have that picture in your head of that mature dominant, that mature follicle? And how many granulosa cells there were? Okay, so lots and lots of estrogen being produced now. Okay, so at some point we have, that is a big arrow, lots of estrogen. Okay, and what happens now is at some point the estrogen levels become so high that instead of a negative feedback to the brain, it switches to a positive feedback to the brain. So what does that mean? Well, positive feedback to the brain is now it signals to the brain and it's causing an excessive amount of a particular hormone to be released. And this really impacts the pituitary gland, and what we see here because of this, lots and lots of LH. We'll see a little bit of rise of FSH as well, but primarily LH that is stored in vesicles in these anterior pituitary gland cells, and all that LH is all of a sudden dumped into the bloodstream, and it goes back to the ovary. Okay, this is called an LH surge. Okay, so large LH peak. This is necessary because this does two things. One, completion meiosis 1, so we form a polar body, and 2, ovulation. Okay? So I think everybody here recognizes that women have a monthly cycle. Okay, so how far are we in the monthly cycle? Roughly. About midway. Okay, so we're only halfway. So if you already thought this was a lot, we're only halfway there. Okay. So what we're going to do from here on, okay, so that's kind of what's shown on the previous slide. What we're going to do now is we're going to add some phases to the monthly cycle, just to kind of explain what is going on in the ovary. So these are ovarian phases. They're divided into a follicular phase and a luteal phase. And this is helpful because these two phases actually describe kind of what's happening. Everything that I just talked about is called the follicular phase. I've been talking about follicles, like growing follicles, about a mature follicle, ovulation. That's all follicle stuff. Okay, so that's the first half. The second half is called the luteal phase, because what happens after ovulation? Do you remember kind of what that, okay, so I don't want to make a beautiful drawing, one, because I can't, I suck at drawing, but two, I don't really have time. But this is a gigantic follicle, a mature follicle, and they had all these cells here, here, here. They had this little stalk here, here's the egg, and some cells around it. Actually, that's not that horrendously bad. It could be worse. Okay. Do you remember what ovulates? So ovulating again means that the wall of the ovary breaks open and something leaves, right? So what leaves from this? The egg and this part, right? That part leaves. And the fluid as well, and we talked about ovarian cramps and stuff like that. Okay, it sounds horrible. I have no idea how bad it can be. You guys tell me. Actually, don't tell me. I should not be asking, let alone I don't even want to know. Anyway, this escapes, okay? This is going, and where is this going? Where does that thing go? What structure?
Fallopian tube.
Fallopian tube, okay? So this leaves, but it means all this stuff, theca cells and granulosa cells there stay behind. And they reform. They actually cluster together, the cells mingle together, and they form what is called a corpus luteum, CL. And the corpus luteum becomes very vascularized. Okay? And this corpus luteum, think of it as a tiny baby endocrine gland within the ovary. And that baby endocrine gland, or the corpus luteum, starts secreting progesterone. Lots and lots of progesterone. Okay, yes?
What part becomes the CL? Is it the follicle or the egg?
Well, we just talked about what happens to the egg. So the egg is not part of this anymore, right? So whatever leaves cannot be part of forming the corpus luteum. But again, I want you to picture, maybe not that crappy drawing, but a mature follicle. If I take that egg and the surrounding cumulus cells out of it, what is left behind? That forms the corpus luteum, right? So the two hormones are progesterone and estrogen that the corpus luteum synthesizes. And especially a lot of progesterone, a ton of progesterone. And this continues through the rest of the monthly cycle. Now, the corpus luteum is very important. Those hormones become very important. So where these hormones go, okay, well, they go all over the place, but one is the brain. It causes negative feedback. Two, it goes to the uterus. Okay, so it's telling two stories. It's going to tell the brain to stop secreting FSH or LH. Okay, why? Why would it do that? Why is that important? One of the hormones that we talked about is called follicle stimulating hormone, right? So follicle stimulating hormone, what do you think it does? It stimulates follicles to grow. So when we just ovulated an egg, and recognizing how valuable these eggs are, because we only have a number of them, do you continuously want to stimulate follicles to grow? No. Okay, so this is an important role for progesterone and estrogen. Stop stimulating these follicles, give them a break. The second story it needs to tell is, what we kind of, how I set up this lecture was, it needs to talk to the uterus. The uterus is the place where, if there is a fertilized egg and an embryo, that's where it's going to implant. That's where we're going to have the pregnancy. So it basically has to tell the uterus, okay, get ready. I've just released an egg. There may or may not be an embryo coming. Okay? Either way, I don't know, but the uterus better be ready. So what are we going to do to the uterus? Well, that inner lining, the endometrium, becomes really thick. Okay? Because an embryo, like when we were about 7 days a week old, we were called a blastocyst. And you know what we were to mom? A cancer. That's really it. Okay? We were 7 days old, we latched onto that thick endometrial lining, and we started invading all that tissue. Okay? Changing blood supply, demanding nutrients, all that stuff. Okay? So that's why we need to have lots of progesterone, because progesterone sets up, and estrogen too, sets up the endometrial lining to be a really great place for an embryo to implant, attach, to grow. Make sense? Now, the corpus luteum is pretty amazing, but in humans it's about 11 days. It can live for 11 days. If it doesn't receive something, like a special signal, then it's going to die after 11 days. That's pretty amazing, right? We're talking about a tiny little tissue or organ that has a lifespan of 11 days. It's awesome. Well, yes and no. After 11 days, when it dies, what happens to estrogen and progesterone? So the corpus luteum dies. It's going to disappear. What happens to all the progesterone and estrogen? Yeah, it goes down. So what happens to the negative feedback of the brain? Yeah, it goes away. So we start secreting FSH again. What happens to telling the uterus to be ready? Doesn't have to anymore. Menses. So basically, menses, okay, is really all that endometrium lining that you've built up, you slough off and you start all over again. And that happens every single month. Okay? Make sense? There's a lot of me talking. I'm going to show some slides now to say the same thing. Okay, so the follicular phase, so you can follow this along. The one note that I want to make, it says here day 1, day 7, day 14, day 25, so 11 days, day 28. Okay, maybe men don't know this here, but women definitely know. Is this exactly correct? No? Yeah, I'm not going to ask you personally, what is your cycle like? I get in trouble for that, but no, okay? For all intents and purposes, just recognize we need to put some time scale here, otherwise I don't know how to explain this stuff, okay? But a cycle could be shorter, a cycle could be longer, okay? A cycle could be irregular, okay? You can have no cycle. That's usually, when do you not have a cycle? When do women not have a cycle?
When you're pregnant?
When you're pregnant, that's one. Anemic, well, that kind of depends. So you can be anemic and start having irregular cycles. You can lose so much weight and have irregular cycles, like those extreme athletes. There's so little body mass. You start skipping cycles, all that stuff. Menopause is another period. You're not cycling anymore, right? So normally, without doing anything else, if you're pregnant or you're in menopause, you don't cycle. Okay? Okay, so this kind of describes what happens in the follicular phase. This describes what happened in the luteal phase. So what I would do for studying for the exam too is kind of think about the big picture. What is happening here constantly? Okay, what hormones are involved? And this should help, the next two slides. So there's two graphs here that focus on hormones. Okay, and it's divided, the dotted line here divides the follicular phase here and the luteal phase there. Okay, so just like I said, FSH stimulates follicles to grow. It's called follicle-stimulating hormone. Okay, estrogens are relatively low. These are small little follicles, so it's not super high yet, but we start making a little bit of estrogen. Okay, negative feedback to the brain, or little negative feedback to the brain, but there is a small amount. Then these growing follicles secrete more estrogen. So if you look at estrogen levels here, they continue to increase. Most of this estrogen comes from once we have selected a dominant follicle or a mature follicle, that one follicle that continues to grow, which happens in about a week into the monthly cycle. Okay, that's going to make more and more and more. Estrogen levels go higher, higher, and higher. See, it peaks here. And then all of a sudden, what happens to LH? Look at this green graph. It's pretty flat. And all of a sudden, this ginormous peak, the LH surge. Okay, and that causes completion of meiosis 1 and ovulation. So now the next slide, we're going to focus on what happens here. Okay, so same graphs here, now just this side. Corpus luteum forms. Corpus secretes lots of estrogen and especially progesterone. Just look at this graph here. Both are high. Estrogen is high, but progesterone is super high. Okay, negative feedback to the brain. Okay, so we're not stimulating the ovary anymore. And then at some point, corpus luteum degenerates. What happens to these? The levels drop off. So now we can have no negative feedback to the brain, and we do this every month, over and over and over again. Okay, so what I just showed you was what happens during the ovarian phases, but it focused on the brain. The next slide focuses, actually the next two slides focus on the uterus part. Okay, but it's the same thing. So because of the ovarian uterine interaction and this rise and fall, especially of progesterone and estrogen, because half a month we have this and the other half you have gigantic amounts of progesterone, it changes what happens to the endometrial lining. So basically the uterus has phases too during the monthly cycle, right? It has a phase where we're going to make something, and then we're going to get rid of something, and we're going to make something and get rid of something, okay? And that's kind of shown here. So on the bottom here, what I like about this figure is it shows the ovarian phases, follicular and luteal. It shows you where you have ovulation, okay, so about 14 days. Again, this varies. And it shows you what happens to the endometrium right here. So the uterine phases are shown here. They're divided into proliferative, secretory, and menstrual. So the follicular phase involves making the endometrium very thick, proliferating, lots of proliferating cells. Once we're in the luteal phase, when we have lots of progesterone and estrogen, we maintain this lining. We make it a great environment. We're making it like the perfect house for a potential embryo, okay, that is going to attach here and invade this tissue and grow, okay. Now, progesterone levels drop because the corpus luteum dies. This lining falls off, and that's what we lose with menses, okay. Now, this does not happen. And progesterone is really important for this. If an embryo implants and there is fertilization and we are pregnant, do we want this to occur? No, that's how you lose a pregnancy. Okay, we cannot lose the endometrial lining. Okay, so anything that interferes with progesterone, okay, and you lose that progesterone signaling will cause a potential embryo to be lost or an abortion. Okay. So maintenance of progesterone levels are really, really critical if there is fertilization, if there is an embryo, if there is a pregnancy. And it actually has to be maintained throughout pregnancy. Okay? Any questions? Good? Awesome. Okay, the next two slides I'm going to do pretty quick. Basically, it's kind of like I did for Sertoli cells. I made a summary slide for the functions of estrogen and progesterone. Some of this we talked about, some other things we haven't talked about yet. Okay, so it will come. So follicle growth, secondary sex characteristics. This is usually at around the time of puberty, breast development, more fat deposition in the hips and the thighs, things like that. Typical of female secondary sex characteristics. The growth and proliferation of the epithelial lining is what we just talked about. It's the uterus, okay? Feedback to the brain and milk production. So in this case, we talk about lactation, okay? Which we'll talk about. Progesterone, kind of the same thing, okay? Key function here to point out is that it inhibits milk production. For estrogen and progesterone, also inhibits milk production. This one is very important. Progesterone inhibits myometrial contractions. Why is that a good thing? You're pregnant. Lots of progesterone. It maintains the lining, so we keep the baby. Okay? And it inhibits myometrial contraction. Does it kind of make sense why that's a good thing? Trust me, when you're 6 months old, you don't, or 6 months into pregnancy, you don't want contractions. Okay? Okay, so this was actually on the little questionnaire that we had, and I was very happy to see that most of you got it. Pretty much everybody got it, I think. Again, so I don't really have to do my little spiel here, which is really nice. In other semesters I do. We know this now, right? Nothing is 100% unless you don't have sex. That's the only thing. If you don't want to get pregnant and you want to be 100% sure that you don't want to get pregnant, then don't have sex. That's it. Otherwise, it is what it is. Okay? You've been informed. So the one that is probably, condoms are not interesting to talk about, that's just a barrier. In terms of reproductive physiology is the birth control pill. Okay, so the birth control pill is usually a synthetic progestin and estrogen. Okay, and they're very potent. They're usually more potent even than endogenous hormones, so they work really, really well. Okay, so how does that work? Why can we use that? I should stop saying we. I don't use it. If you're using that, why does that work? Although not 100%. But why does it generally work?
Tricks your body.
It tricks your body. Okay, explain. Like tricks a woman's body how? Always damn these follow-up questions.
Basically, it changes your hormone levels such that either ovulation or implantation are not supported.
Yeah. So so we talk about two hormones, right? Estrogen, progesterone. During the ovarian phases, what are we talking about? What is this characteristic of? The follicular or the luteal phase? During which of these do we have most progesterone? Luteal. Okay. What does progesterone do? Inhibits the brain. So if it inhibits FSH and LH, can we still have follicles growing or ovulating? No, so basically it's that. You prevent ovulation. There's also some effects on the thickening, the mucus of the cervix and things like that. But that's really the key thing here. Inhibits LH and FSH release, prevents ovulation. Okay. Okay. Now we know exactly how men make sperm. Now we know exactly how women go through their monthly cycle and have an egg released. Since we're talking about reproductive physiology, the goal is to get these two together and make a baby. Okay? So you can follow. Yeah.
It prevents menopause.
Oh, yeah. Sorry. Yeah, thank you. Yes, so, okay, so now that we know how the birth control pill works, can we not expand, what is delay menopause, or even prevent menopause? Right? I mean, I just shut off the brain, stop telling the ovary to keep growing these follicles. I want to wait until I'm a little older. I want to save all my eggs. What do you think? Is he a no? Anyone yes? Some people are a little like, I have no idea what to think here. This sounds like a trick question. Some people thinking, why? We don't know this yet? Well, one thing to understand is, okay, when these oral contraceptives came out, that wasn't 100 years ago, okay? So really, if you wanted to really know whether this is true or not, you need to do this in a very large scale. But how would you do that? How many people would you possibly have to recruit for this? First, we don't even know when a woman normally would enter menopause anyway. Some women at 50, some women at 60. So how would you know when you're delaying menopause? So it's a very practical problem. But just let's hypothetically say that every woman hits menopause at the age of 50. Okay? Now we have somebody that decides at the age of 15, 16 to take birth control pills. Okay, that's the second group. Okay? And they're going to be on there for 20 years. Then they say, you know what? Now I'm going to get off of that and see. So 20 years later, so that means they should hit menopause when they're 70, right? No? So I'm very skeptical. Why not? Doesn't it sound good? What are we missing? Okay, go for it.
Well, actually, there is.
Yeah.
The follicles, they have a stage of maturation that they are. They don't depend on hormones.
Mm-hmm.
So the first stage of maturation, so they don't need FSH. So they start maturing, and when they are mature and they develop these FSH receptors, then is they are sensitive to FSH.
Mm-hmm.
So in our ovaries, we have many of those, and usually in waves. And the FSH that comes, those are the ones starting. When we are in birth control, all these are maturing. So usually what it happens, if they are waiting there for long until stay, some start going into atresia. But if you stop taking these pills, this is when it happens that sometimes you ovulate multiple oocytes because there are many waiting there.
Mm-hmm.
So you can stop that maturation with birth control. So your follicles are going to or ovulate when you stop having, so maybe you ovulate 5.
Yeah.
Or they're going to go into atresia. And it's impossible to prevent that part.
Yeah.
Is twins multiple eggs being released?
Yeah, we'll get to that in a minute. So just to follow, so for this part, atresia, cell death. Okay. So what happens is, you remember, we didn't really talk about it. I'm not sure if Dr. Warren talked about it. He probably left it for the repro part. The cells in the pituitary gland that release LH and FSH are called gonadotropins. Tropins means nourishing, okay? So LH and FSH are nourishing hormones, okay? And if these somewhat maturing follicles that initially grow, they don't even need FSH for any of that. If they start growing, at some point, they need nourishing hormones. Otherwise, you die. Okay? So basically, whether you are, if you're on birth control pills or not, cells are dying, follicles are dying all the time. We saw that on the graph. Okay? So just by preventing ovulation, okay, doesn't mean there are no things that are dying. They're going to die off. And like Dr. Parsons said, if all of a sudden you do get off of the birth control pill, you start, there's a whole bunch that are sitting there, they'll start ovulating a whole bunch of them. Okay? But you lose more. Okay? The whole point is you have a fixed number, which is somewhat different for every woman. And you're not keeping that number by merely inhibiting LH and FSH release. Does that make sense? So it's a very good question. This is what we're assuming because, honestly, practically, we don't know. Because you can't really study this practically in humans. You would have to have millions of women starting at the age of 15, tell them to be on birth control pill consistently all the time for like 5 or 10 years, and then tell another million plus women to not take any birth control pills, okay, and follow them all the way until they hit menopause. Well, how long is that study going to take? Well, it's more than my lifespan, so who's going to do this? This is just not practical, okay? But it's a good question nonetheless, okay? Okay, fertilization. We're going to get kind of into something that came up in, I think it was one of the questionnaires. I forgot the question. Anyway, what we have on the left side is the end result of ovulation. What you see there is an oocyte. And how should I call that oocyte? A primary oocyte or a secondary oocyte? Why do I call it a secondary oocyte?
It's going through meiosis 2.
Yes, it's entered meiosis 2, but it's arrested again in meiosis 2. The other thing that you see is kind of that fluff around it, right? Those are those cumulus cells that are kind of embedded in this matrix, okay, that came out of the follicle, okay? But now the cumulus cells have kind of expanded. They're not stuck together anymore, but they kind of held to this gooey matrix, okay? So this is what we find in a fallopian tube, okay? That egg is viable for about 48 hours, about 2 days. That's it. On your right side is a cartoon picture of a sperm. Okay, that thing is viable for about 72 hours, so 3 days. Okay, so I think one of the questions on the questionnaire was, how did I word that? How many days during the monthly cycle can a woman get pregnant? I think that's how I worded it. Right? Yeah. So some people said 28. Well, that would be something if you can get pregnant 28 days out of your 28-day cycle. That is too much. Okay? So if you kind of think about this stuff, so how many days out of the reproductive cycle? Approximately, okay? We're saying these numbers, but there's some variability there. How long is an egg alive for? 2 days? How long is a sperm alive for? 3 days? If they merely just overlap at the right time, the egg is barely holding on, and the sperm is just same thing, and they yet get together, right? So the maximum period will be about 5 days. Make sense? Okay. I don't know if there was anything else on the questionnaire. Am I missing one thing? I don't remember. Okay. So anyway, that's it. Okay, we need to get these things together, okay? And so we need fertilization. So this is a good depiction of the female reproductive tract. It's missing the ovary, but that's okay. We kind of know where the ovary should be here. There you go. Ovaries are right here. I love this. Whose is this? Abby's. This is perfect. Okay, now that we've had female reproductive physiology, what am I holding here? Oh, please, somebody say something. Come on. Yeah, okay, be very specific. My fingers are touching maybe the cervix or the uterus, fallopian tube, et cetera, Okay, so as you know, for reproductive purposes, the sperm gets deposited in the vagina during intercourse. Okay, but it's going to see it needs to go all the way up the female reproductive tract and find the egg. As I told you, the egg is ovulated, goes to the fallopian tube. So fertilization takes place in the fallopian tube. That's it. Okay, so it needs to go through two important barriers. Okay, and the barriers to fertilization, the first one is the cervix. Okay, so the cervix has a lot of folds. Okay, it can have a lot of mucus and a lot of different smaller grooves and larger grooves, and in fact most of the sperm, so again an average ejaculate, let's say 250 million sperm, most of those will never get through the cervix. Especially if there's some abnormality with the sperm, like maybe the tail is not working or it's kinked or there's something wrong structurally, it's never going to get through the cervix. Okay, so now it made those few that made it through the cervix are now in the uterus. Again, remember the sperm are not swimming. Right? It's smooth muscle contractions from the uterine walls that kind of help move the sperm along. Okay? Now, how many eggs are we ovulating on average, normally? 3, 4, 5, 6, 7? 1? How many? 1? Yes, 1. Okay, 1 egg. Which side? Well, yeah, so we can divide the sperm up in smart sperm and stupid sperm. Okay? The smart sperm pick the right side where the egg actually ovulated. The stupid sperm go the wrong way, so they never actually make it. Okay, so but that gets us to the second barrier, and that is called the uterine tubal junction, or UTJ, uterine tubal junction. Yes?
Is which tube they pick like random?
Yeah, that's not really sure how they know which one to pick. Okay, once they're in the right fallopian tube, there are some signals. Some of it actually includes progesterone that is in the follicular fluid, like that was in the follicle, because that fluid actually, some of that goes with the egg into the lumen of the fallopian tube. So some of the progesterone there actually act on the sperm, and we'll do something to sperm that we'll talk about in a minute. Okay. So the uterine tubal junction is the second barrier. The uterine tubal junction is the fallopian tube connecting to the uterus. Okay, so that's the stuff right here. This is a uterine tubal junction. That's a uterine tubal junction. Okay, second barrier to fertilization. And then finally, this would be a cross-section of what a fallopian tube looks like. And there are lots, it's more granular, okay? There's lots of things in cilia, lots of places for the sperm actually to get stuck, okay? And again, sperm there are alive for 72 hours. So time is somewhat of a factor if we want fertilization to occur. Okay, and then finally, if the egg is ovulated here, well, fertilization will happen right there. Okay, so one of the things that happens, so this is 2 slides before we have a break. Okay, we talked about sperm maturation in the epididymis, right? When it was at the head of the epididymis, those sperm couldn't even swim, even if you begged them. Okay, they would still not swim. They're just physically impossible to swim. After they have been in the epididymis for a while and they're in the tail, now they do have the ability to swim. If you give it the right stimulus, they will start swimming. Okay, so there was kind of a maturation for sperm there. The second part of maturation happens in the female reproductive tract. And what I want you to know is the word capacitation. Capacitation means final maturation of sperm in the female reproductive tract. So this occurs in the female reproductive tract. And capacitation involves 2 things. First, we're going to increase sperm membrane fluidity. Fertilization involves 2 cells with a cell membrane having to fuse. I know Dr. Warren at the beginning of the class talked about membranes and what's in a membrane, phospholipids and other proteins and things like that. And you could make a membrane really rigid or you make it more fluid. What is that one molecule that you can either put in a cell membrane or you can remove it that affects fluidity of that membrane? Cholesterol. Cholesterol, exactly, cholesterol, okay? So basically what we're doing here is that increase in membrane fluidity means that we're going to start stripping out some of the cholesterol out of the sperm membrane, make it a little bit more fluid, so it's easier for fertilization, okay? The second part of capacitation or sperm maturation is a hyperactive motility. Hyperactive motility. This is the part where the sperm really starts beating its tail and really starts swimming. And this happens when it's really close to the egg. Okay, so the flagellum starts beating like crazy. Okay, and basically it's trying to swim and really get to the egg first. Okay, and once it gets to the egg, okay, do I have this one? Okay, here. This is kind of a crappy picture because I just showed you that these cumulus cells are not like a brick wall. They're more expanded, right? But this hyperactivation really helps for the sperm to kind of go through all that gooey stuff. Okay, does it make sense what I said about capacitation? The two things that involve capacitation? Yes? Good. Okay, so now the sperm undergoes capacitation. It does all that stuff. It's going to go through this whole layer. Okay, and at this point there may be 20 to 200 sperm left from the 250 million that we started with. Okay, so now it's a foot race. The first thing, it's going to go through all these cells and it's going to hit that structure that we call the eggshell, the zona pellucida. Okay, and then the sperm touches the zona pellucida. You have to know this, when the sperm touches the zona pellucida, it causes an acrosome reaction. Remember when we mentioned yesterday that a mature sperm, in the head of the sperm we had the nucleus and we had this bag of hydrolytic enzymes, the acrosome. And basically what we're doing right now is the acrosome reaction is that bag with enzymes. It's going to burst open and all these hydrolytic enzymes are being released. And these hydrolytic enzymes are going to start eating or drilling a hole through the zona pellucida. And the sperm is beating its tail. It takes about 5 to 10 minutes for the sperm to actually go through the zona pellucida. The tail is beating like crazy. Okay, and then finally it gets to a little bit of space that is right here between the egg membrane and the zona pellucida. And now the sperm can fuse with the egg. Okay, does this make sense? Okay, time for a break. 5 minutes? Good. So many more exciting things to talk about. And there's still a couple of things that I need to really cover before I let you guys loose and deal with the exam tomorrow. Okay, so we just talked about, so again, understand what capacitation is, okay, what it means. Understand that hyperactive motility, which was the second part of capacitation, then involves hyperactive motility and we start swimming. Okay, and we start swimming through the cumulus cells. We start getting into that space here a little bit, but the zona pellucida is not shown here. And we start fusing with the egg membrane. Okay, and that causes completion meiosis 2. The sperm has to fuse with that oocyte membrane, fertilization, for meiosis 2 to be completed. And so that also means we're kicking out what? A second polar body. Okay, make sense? So, it is in mammals, other species do different. In mammals, it is very important that we only have one sperm fusing with an egg. If there are 2 sperm fusing with an egg, or 3 or 4, that is never going to develop into anything. Okay? Some species you can actually have no sperm fusing and the egg still gets activated and develop. That's called parthenogenesis, but that is in other species, not in humans. In humans, that never works. Okay, so that also means if we have like 20 or let's say 100 sperm right there, they all undergo capacitation. They're all going to start doing this thing. So what prevents another sperm from getting there at the same time, or just right behind it from fertilizing the egg? So there's something called a block to polyspermy. Polyspermy meaning fertilization with many sperm. There has to be a block to that. Okay, and that's kind of what's shown on this slide here. So when we start here, many sperm bind to the receptors on the zona pellucida. It's kind of like a receptor that causes the acrosome reaction, that causes the enzymes to be released and drilling a hole through the zona pellucida. Okay, sperm move through the zona pellucida. It takes about 5 to 10 minutes. That's trivia. That's not on the exam. And then one sperm binds to the egg. It kind of binds to the egg and they start fusing, okay? And then the first thing that happens is a change in membrane potential of the oocyte. So that's the first part of a block to polyspermy. You change the membrane potential of the oocyte. If there's a sperm that's right behind it that still tries to fuse, it cannot do so anymore because of change in membrane potential. Okay? The second thing that happens is something called a cortical reaction. In fact, the oocyte on the outside, so this is an egg now, okay? Well, it's a circle, but an egg. There's a nucleus somewhere. There's a whole bunch of vesicles on the outside. It's kind of like an acrosome, but like lots of baby acrosomes. We don't call it an acrosome, we call it cortical granules. But these are little bags with enzymes. Okay, so when the sperm fuses, all these enzymes are being released. It goes to the zona pellucida, that eggshell, and makes it extremely hard. Okay, so it starts cross-linking things even more. It makes it like an impossible thing for sperm to go through. So sperm actually, if they're like, it takes like 5 to 10 minutes to go through it, right? So if they're kind of halfway through, and that stuff happens, they're going to get stuck. They're not able to get through that stuff anymore. Any sperm that's on the outside is not ever able to get through that stuff anymore. So does that kind of make sense, the block to polyspermy, those two events? Okay. So then, so zona hardening, fewer receptors, we get completion of meiosis 1, pronuclei, etc. So the pronuclei, the reason why we call it a pronuclei is a pronucleus has 23 chromosomes, a normal nucleus has 46 chromosomes. So the sperm has a pronucleus, 23 chromosomes, and the egg also forms a pronucleus after completion of meiosis 2. So you have a male and a female pronucleus, each has 23 in that egg. And so that those pronuclei kind of have to fuse, and they already start dividing their material to start embryogenesis. Okay, yes?
So when the egg kicks out that second polar body, how does it know which set of chromosomes to kick out?
It's random. So they are all aligned. So we do have all these butterflies, right? Because that's meiosis 2. We have 23 of those things. And that needs to separate. But that is a random thing. So they're all lined up like that. We have genetic swapping, so it's not identical anymore to what mom was. And so everything gets lined up. The spindle pulls it apart, collects all that stuff, puts it in a vesicle, and kicks it out. Okay? Okay, and then activation of enzymes, embryogenesis. Okay, we're going to do a little bit of developmental biology now. Okay, so basically what we have here is the female reproductive tract, obviously. Here's the ovary, an ovulated egg. Sperm swim, there's only 3 here. That's kind of sad, but let's just pretend there's more. And one of them fertilizes the egg. Okay, and so day 1 fertilization. The first, once an egg is fertilized, we call that a zygote. Okay, so zygote. That is a fertilized egg. That is 1 cell, 46 chromosomes. We all were a zygote. Okay, that had a potential to develop into what we are now, which consists of about 37 trillion cells. Okay, with organs and tissues and all that stuff. All coming from that 1 cell. It's amazing, right? That is a true stem cell. Okay, so as it starts dividing, that 1 cell zygote becomes a 2 cell, 4 cell, 8 cell, 16, 32, 64, 128, I think you get the gist. Up until there's something called a blastocyst. This is a blastocyst. That blastocyst is what I mentioned earlier. We were a blastocyst after about 7 days, 5 to 7 days. We were that cancer. Okay, blastocyst is a cancer. It's going to attach to the endometrial lining right here. And see what it does? It starts invading. Okay, that's the pregnancy. Okay, think of the timeline here. Ovulation. Think back about where we are in the female reproductive cycle. We ovulated an egg. Sperm got there. Fertilized it. It takes about 7 days for that embryo to get to the endometrial lining and attach an implant. So this part, what we're describing here, which of the phases are we from the ovarian phases? Where are we? Are we in the follicular phase or in the luteal phase? Luteal phase, right? It's after ovulation. How long so this is a critical time, right? Stuff has to happen here. Because we have 11 days during the luteal phase where the corpus luteum is alive and secretes progesterone and maintains this lining. Okay? How long does it take for that embryo to get to the endometrial lining and attach? 7 days. So it's getting awfully tight, right? We don't have that much time left. What needs to happen? The embryo has to tell mom that, hey, I'm here. Okay, keep the corpus luteum alive, please, because without it, I'm going to disappear with the endometrial lining. That is called maternal recognition of pregnancy. Okay, so the baby is actually, well, baby, the fetus, the embryo, is communicating to mom. It will send out a signal and tell mom, I'm here. That signal is actually a hormone that actually will go to the ovary and it will keep the corpus luteum alive. So the corpus luteum stays around. Instead of dying after 11 days, it stays around and it keeps synthesizing progesterone. Yes?
So when you get pregnancy test, what is it looking for?
It is that hormone that I just mentioned. It actually detects that maternal recognition of pregnancy hormone in urine. Because that hormone will actually enter the bloodstream, and you know how the kidneys work now. You guys had an exam on that. That was fun, right? The renal physiology is, oh, I love kidneys. Come on. Those 2 little thingies that do all that stuff, filtering all that blood and plasma. Anyway, okay, so that stick, when you pee on it, it's going to detect the presence of that maternal recognition of pregnancy hormone, okay? Okay, so what happens? We have fertilization happening right here. And that embryo, all of a sudden, bloop, falls out. So normally what happens, see these fingers here? They're called fimbria. When ovulation happens, those fingers wave over the surface of that ovary and actually capture it. So it's not like the ovary has to shoot it out with so much force, and it's actually like target practice, like where's the opening of the fallopian tube? Ah, got it. No, it's not like that, okay? So it actually waves over and it captures that ovulated egg. But let's say there's a mistake, okay? And instead of that embryo going nicely down the fallopian tube, see, it's not getting bigger, right? The cells are getting smaller. Otherwise, it gets stuck. But it goes all the way here through the uterotubal junction, and it's fine. Oh, but it makes a mistake, goes the wrong way. Bloop, and it's out. What happens?
Is that ectopic pregnancy?
Yes, an ectopic pregnancy. Now, this kind of ectopic pregnancy is extremely rare. Okay, because basically this means we have now an embryo that is floating around in the peritoneal cavity near the abdomen, and it's going to attach somewhere, like it does something, okay? And then it's going to try to develop and get nutrients. The only way it's going to grow if it latches onto some blood supply or something, okay? So these are ectopic pregnancies. They're extremely rare, but they can lead to something called a lithopedion. Okay, so basically there's certain variable degrees of development, but in most cases these just die off and become calcified or stone. They're referred to as stone babies. Okay, some cases they develop pretty far along. Like if you could see this one figure on the top, this is a lithopedium. This is a baby with a head, limbs, body, but it could not be sustained, and the body just takes care of it by putting a lot of calcium on there. So it calcifies that baby, and that woman doesn't know that she's pregnant. Here you can see organ development. There have only been, in some very obscure journals, a publication that where they indicated a successful pregnancy through an ectopic pregnancy in the abdominal cavity that actually led to a live offspring, okay, usually through a C-section. But that is extremely rare. Most of the ectopic pregnancies that happen because basically the embryo starts implanting or attaching too soon, and it actually tries to implant it to the fallopian tube, the wall of the fallopian tube. And that is extremely painful, causes a lot of bleeding. And so you either, if it's caught really early on, you could give a hormone treatment to get rid of it, and otherwise it has to be removed surgically. Okay, so that's about 1% of the pregnancies in the U.S., I believe, are ectopic pregnancies right here. Okay, a little bit of embryology, I guess. Okay, so these are all human embryos. Okay, I want to focus on a couple of things. This one right here, you should be able to tell that that is a zygote, a fertilized egg. The two hints are, okay, hold on. This is the zona pellucida. This is the oocyte. There are two pronuclei. The only way you can have two pronuclei is if you have a fertilized egg. There are two polar bodies, 1, 2, right there. So this is actually what you can monitor under the microscope. So for example, in IVF clinics, if you want to see if you've actually successfully fertilized the egg, all you do is put egg and sperm together and monitor under a microscope where you have that. Okay. Now, 2 cells, 4 cells, lots of cells. This is called a morula, a compact morula. Basically, you cannot see the individual cells anymore. And then finally, that develops into a blastocyst that's going to implant. And implantation is nicely shown on this slide right here in yellow. So it could implant here, 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 okay, all over the place. Okay. So the things I want you to know about the blastocyst is that it is comprised of 2 different parts. Okay. So I'm going to cheat a little bit. So now this is not an egg anymore. Okay. Now this is a blastocyst. Okay. So the outside layer is a single layer of cells, okay, a single layer, and that is called the trophectoderm. And the cells are called blastomeres. No, actually, no, trophectoderm cells, just leave it. Trophectoderm, those are trophectoderm, okay? That is going to give rise to the placenta. So the trophectoderm actually gives rise to the placenta. This clump of cells is called the inner cell mass. The inner cell mass gives rise to everything that you are right now. Okay? Every single organ of your body. Okay? So when people talk about embryonic stem cells, they're talking about these cells that are culturing in in a dish. Those are embryonic stem cells. You could take these cells and you can force them to differentiate in every imaginable possible cell type that makes up our body, except the placenta, because the trophoblast cells do that, or the trophectoderm does that. Okay? So this slide actually shows the inner cell mass right here. This is the trophectoderm, or trophoblast cells, single layer, and it's actually this layer underneath the inner cell mass that starts invading. These cells are very invasive, and in fact, in cell culture conditions, they behave like cancer cells. They migrate, okay, they invade tissues, things like that. Okay, maternal recognition of pregnancy. I just told you that this has to occur. We have a window. There's 7 days that it takes for the blastocyst to get there, and we're competing against the 11-day lifespan of a corpus luteum. Okay? So that hormone that actually is secreted. Human chorionic gonadotropin is the hormone that is the maternal recognition of pregnancy, and it's actually the trophectoderm cells that secrete that. Okay, so they actually secreted hormones. So it's the cells that ultimately will be the placenta. They secrete the human chorionic gonadotropin. So on a pregnancy test, the little stick that you pee on, it's looking for HCG. Okay, and HCG is exactly like luteinizing hormone, except it's way more powerful. There's like post-translational modifications, so it becomes really glycosylated. It's very stable in blood. Okay, but for the rest, it acts just like LH. Okay, and that acts on the corpus luteum, and instead of dying, it stays around. Make sense? So now we can continue to have high levels of progesterone and estrogen, which we need for the endometrial lining. Okay, does it make sense? Any questions? A lot of things happening, right? It's pretty amazing. Yeah.
You said that acts on the corpus luteum?
Yes.
That hormone?
Yep. Yep. It acts on the corpus luteum. Good. And then we have pregnancy. How long is pregnancy in humans? 9 months. I've learned over the years not to assume anymore, so I'm just going to ask. Thank you. 9 months, yes. Okay. And so basically, I told you, throughout pregnancy, we need to maintain or have high levels of progesterone and estrogen. We need both. But progesterone levels always need to be higher. Remember the one thing that progesterone did during pregnancy that was kind of important? It inhibited something. If it didn't do that, you would lose the pregnancy. What happens at the end of pregnancy? Women go through labor. What is labor? There's one word that I'm looking for. Starts with contractions. Progesterone inhibits myometrial contractions. So that's good, right? This whole 9 months, we have all this progesterone that maintains the lining, inhibits myometrial contractions. Perfect. Okay. Now you also see that at some point, again, the trophectoderm cells, the outside layer, secretes HCG. That is, HCG is this green line. Okay. So we need that HCG to maintain the corpus luteum. So all this progesterone estrogen comes from the corpus luteum. At some point, we don't really need HCG anymore, or not even the corpus luteum for that matter, because at some point these trophectoderm cells have developed into a placenta. And the placenta as an organ will start secreting progesterone and estrogen. Okay? So after the first 3 months of pregnancy, all the rest of the pregnancy, all the progesterone and estrogen comes from the placenta. Okay? So the placenta is my favorite organ ever. One, because we study it in the lab. And two, it's a temporary organ. And it's pretty amazing what it does during those 9 months. Okay? It's also one of the most difficult things to study, because when you're pregnant, how many pregnant women are willing to give up their placentas? Yeah, none. So basically we get the stuff after pregnancy when it's tossed away. Okay? Trying to prevent women from eating it and stuff like that. I'm not going to go there. I don't have enough time, but there's lots of fun stuff about placentophagy and things like that. Just a quick note. Do whatever the hell you want with the placenta, okay? You want to eat it, go eat it. You're not going to die from it. But if somebody tells you that it's healthy and nutritious, that is bullshit, okay? There's absolutely not a single nutritional reason to eat your placenta, okay? But go ahead, it's all fine. Companies will put it in pills for you because it looks a little nasty, okay? Yes?
Really? Is that a common thing that people do?
Well, it's more common now because I don't know what's happening the last couple of decades, but people are starting to become crazy, okay? We're starting to look for all kinds of solutions for not feeling happy or whatever. I'm going to get a little sidetracked here, but...
Eating the placenta is how you get happy again? Or what?
No, but people, we call something like that a placebo effect. Okay? Yes, you can tell women can, or couples can send their placenta to a company, they'll boil the shit out of it, pulverize it, put it in a capsule, and you eat it. And if you ask those women or men if they both eat it, does it make you feel better? Well, sure, if you think you're going to feel better from it, then you're going to feel better from it. Okay? You're not going to lose weight from it. It doesn't do anything. Okay? It's the placebo effect. You can also toss it in a yard and plant a tree. The tree will grow anyway. Okay? It doesn't matter. But
Does that provide calories?
no, Well, nothing. It provides nothing. If you're a little anemic, because the idea is it's very vascularized, a lot of blood and stuff like that, yeah, how about you just take a little iron pill for that, okay? That will work just as well, okay? Anyway, I need to get off of this because I could talk about an hour about this stuff. But the placenta, it's semi-permeable, okay? Yes?
Could you tell us what it is that induces labor?
Yes, we're going to talk about that. Yeah. So the placenta, semi-permeable. It needs to provide nutrients and oxygen, and it needs to remove waste and carbon dioxide. Okay, so it has to be semi-permeable. It has an endocrine function. We talked about that. Progesterone and estrogen are critical. There can absolutely not be any mixing of blood. Fetal blood and mom's blood can never mix. That will cause the pregnancy to fail. And this makes sense because the fetus is not exactly the same as mom. We've gone through meiosis. Okay, the fetus is its own genetic identity. So blood can absolutely not mix. Okay, and basically what it looks like here is this portion. This is the endometrium. There's a lot of glands, a lot of blood flowing into this. And here is that chorion, the trophectoderm cells. So we don't call it trophectoderm cells anymore. We call it a chorion. Just give it a different name. Okay, and these chorion at certain places have all these tree-like structures. Those are called chorionic villi. Okay, and basically what separates mom's blood from mom's blood is all this purple stuff here, and fetal blood, which is right here, are those chorionic cells that are right here on these villi. Okay, and in some species it's a single cell layer, that's all. Okay, in other species a couple of more cell layers, but basically that's what we're having here. Okay, labor, we had a question about labor, regulation of contractions and all that stuff. So what happens? Well, we have 9 months of pregnancy. At first, you don't see it, so you just, never mind. You're pregnant, and the baby starts growing, and it gets bigger, and it gets bigger, and it gets bigger, and now we're at 9 months, and it looks ginormous, and you feel almost sorry. It's like, how is this possible? It's amazing, okay? So I hope you can imagine that at some point that fetus is going to drop more, okay? And it's going to push on the cervix. And that starts initiating a whole cascade of events, okay? First, we're starting to have some cervical stretch. The cervical stretch itself causes a couple of things. One, it is sensed by neurons that signal to the posterior pituitary gland. Finally, the posterior pituitary gland does something again, and it releases oxytocin, another peptide hormone. Oxytocin goes to the uterine wall, okay? And causes the release of prostaglandins from the uterine wall, and prostaglandins cause uterine contractions. Oxytocin itself causes uterine contractions. The only thing that I want you to put in here as well is that estrogen. So estrogen is not on here, but where this arrow, can you see the arrow? Oh yeah, there. Put estrogen here. Estrogen actually also promotes prostaglandin release from the uterine wall. Okay, so basically we have oxytocin, prostaglandin release, they all cause uterine contraction. Now, if you're standing, you have this baby right here, it's starting to push down on the cervix, and I do endometrial contractions or uterine contractions. What's going to happen? Well, squeeze the baby more on the cervix. You put more cervical stretch. Okay, so you're going to do this over and over and over again. Okay, so they become more progressive, more regular, things like that. Okay, progressively stronger, more coordinated, directional as the labor progresses. Okay, so I just mentioned estrogen a little bit. So finally, the baby is let's do the first part first. So during labor, so this is delivery right here. Delivery means the baby is out. Okay, how come progesterone and estrogen levels drop like that? Why is that?
Because does the placenta also leave?
Yeah, the placenta also is expelled. So if the placenta is gone, no more progesterone and estrogen. Okay. Now, but up until delivery, progesterone levels are always higher than estrogen. So how can estrogen still have some effect on uterine contractions? I just mentioned through prostaglandin release, right? It's because the idea here is that we have something called functional progesterone withdrawal. It doesn't mean that progesterone is gone, but it means that the progesterone receptor is not working as well anymore. It's been there for 9 months. Think of it as just getting tired. okay? I'm tired, I can't do it anymore, I'm done. So slowly but surely, the inhibitory effect of progesterone on the contractions diminishes. And that's how estrogen can kind of skip in there and help with these contractions. Okay, lactation. Okay, so a little bit about estrogen and progesterone. It inhibits milk production, things like that. During puberty, estrogen increases duct growth, branching. Progesterone promotes alveoli growth. During pregnancy, estrogen, progesterone, and prolactin all promote breast enlargement. Okay, but we have no milk production. Okay, what we need, so when we think about lactation, there are two things. There is milk production, and there is milk secretion. Okay, one is making milk, the other one is for the milk to come out. Okay, and prolactin stimulates milk production. Oxytocin stimulates milk secretion. So the way this works is that, for example, the baby crying or suckling or nursing stimulates mechanoreceptors on the nipple. There are neuronal inputs to the hypothalamus. And basically, the hypothalamus starts secreting something. Ignore this, you can put a big X through that. But basically, there's a signal to the anterior pituitary gland to release prolactin. So prolactin, I want you to know, it comes from the anterior pituitary gland. Prolactin acts on the gland cells in breast and stimulate milk production. At the same time, that stimulation through mechanoreceptors also is sensed by posterior pituitary gland, releases oxytocin, causes milk ejection. Okay? So anterior pituitary gland, prolactin, milk production. Posterior pituitary gland, oxytocin, milk secretion. Okay? Okay, perfect. We have about 15 minutes left. I always seem to run out of time. I wonder why. So ambitiously, I've called this last part assisted reproductive technology, stem cells, and cloning. This is overly ambitious. I really want to focus on just a couple of things here, okay? Because as far as I'm concerned now, you guys are experts in reproductive physiology, right? You know everything there is to know. Do we feel more comfortable about the female reproductive cycle? A little bit? How easy was the male reproductive thing? That was easy too, right? Okay, so now let's see if we can apply some of this stuff to real life. These are not odd things, okay? So there's some outline, kind of ignore this stuff, okay? The part that I really want you to kind of learn is something called in vitro fertilization. That's really the part. So as you know, and again, I'm going to put a footnote here. There's a graph here that talks about fertility in women. And what this graph is telling you, that as you get older, your fertility declines. Okay, let's first off, I know there's something called a biological clock, all that stuff. Yes, that is very true. The last thing I want you to do is look at this graph, run out of here after the class is over, and feel the need that you need to just reproduce right now because time is running out. It's not like that, okay? Everybody here knows, if I were to put a question on the exam that is worth 100 points, there are two women, okay? They're identical twins. But that's impossible. But one is 20 and one is 50. So they cannot be identical twins, right? But for the rest, everything is the same. Which one is more likely to become pregnant? The one that's 20 or the one that's 50? It's worth 100 points.
The 20 one?
Who says 20? Raise your hand. Anyone that picks 50? Thank you. Okay, so you already knew this. I could have asked this question 3 days ago, last week, and you knew this. So there's really nothing new on this slide, okay? So the thing with this stuff is people always wonder why and how. Who knows? There's so many things involved. The one thing I do want you to recognize is kind of the biology part of this. Remember arrest and meiosis. So a woman that is 50 years old and hasn't hit menopause yet and ovulates an egg, right? That means for the first time, those chromosomes got to separate. How long have they been arrested in meiosis 1 when she's 50? How long? 50 years. For 50 years, all these hormone changes, the environment, toxins, everything. And you've asked that cell to stay like that properly for 50 years. Okay? And now it finally gets ovulated, separates. So now you're counting on the separation of chromosomes to happen correctly, right? And then fertilization. That's still, all that stuff still needs to happen. Okay? So what we'll see is the spontaneous abortions and the decrease in fertility. In most cases, it has to do with chromosomes, chromosome abnormalities. Okay? So that's really kind of part of it. Does that mean that a woman that is 50 years old, can she not get pregnant normally through natural conception, everything? Yes, you can. Of course you can get pregnant. Okay? There are lots of 20-year-old women that cannot get pregnant. Okay? So don't get too hung up on this something that you already knew anyway. Does it make sense? Okay. Now what we have noticed a lot more is couples that are relying on assisted reproductive technologies. Okay? In order to have a baby or child. Okay? If that's what they would choose. Okay, obviously there are other options available. What's another option aside from assisted reproductive technology if you want kids? Adoption, exactly. Okay, but I want to focus on the reproductive physiology part here. Okay, so we have artificial insemination, hormone-assisted ovulation. The one that I want to focus on is in vitro fertilization. Okay, so the first baby, IVF baby that was born is Louisa Brown. Okay, 1978. Here are the parents, Leslie and John. I think his name was John. Yes, John. Leslie and John Brown. So this was in 1978. Okay, this was the headline. Test tube. Well, if anything, we're not using test tube, but fine. Super babe. Okay, this is nonsense. This is just terrible media stuff. And obviously this caused quite a lot of commotion, uproar and things like that. There were people that were super excited. There were lots of people that thought this was the end of the world. And the amount of hate mail that the parents got for this, it's unbelievable. Blood splattered letters and all that kind of stuff, needles, knives, everything. Okay, recognize that there are hundreds and hundreds and hundreds. I don't know how many couples now have received kids because it was through IVF. Okay, there are hundreds and not thousands of IVF babies. Okay, but this is kind of the timeline. So we're going to play like we're a fertility clinic. Okay, so I'm gonna, because I know there's one person here who worked in a human IVF clinic. So in fact, she should just do this lecture part for me. She's cringing right now and hiding back there. So what I am going to do as a kind of a footnote, I'm going to overly simplify some stuff. Okay, I want to use this so you kind of start thinking about what you've learned in terms of the hormones and things like that. Okay, so in vitro fertilization refers to that in a dish, we're going to put sperm and egg together. We're going to monitor development. And then at some point, usually the blastocyst stage, we're going to transfer that embryo and we're going to put it back into mom. And then see if pregnancy continues. Okay? So the first thing is egg production needs to be stimulated by hormone therapy. Okay, you're all experts now. You're a clinician working in this clinic. What hormone are you injecting this woman with to stimulate egg production? And all of a sudden it hits you. Wow, we've talked a lot about lots of hormones. What do I pick? Okay, what hormone? So estradiol, FSH. You mentioned FSH. Why FSH?
It stimulates the follicles.
Follicle stimulating hormone. That's exactly it. Let's not overcomplicate things. FSH. So FSH analogs can be used, okay, to stimulate follicle production. At the same time, other hormones will be applied too, because the one thing you don't want to have happen is basically you want to control ovulation. Okay, but we're going to focus on just a couple of parts here. So let's pretend there's a whole bunch of follicles, more follicles that are developing. And now we don't want one to ovulate, we want lots of them to ovulate. We're going to do like super ovulation. So how do we get these eggs out of the ovary? What hormone would you provide this woman? If that's the way you wanted to do this. Yeah, usually what they do, they're going to add something that acts on the GnRH, on GnRH itself. But let's just say if you wanted to provide a hormone to this woman to get the eggs out. I mean, you can also go in laparoscopically and retrieve the eggs, but let's just pretend we're giving the woman a hormone in order to ovulate these eggs. What's the first hormone you think of when you think about ovulation? What hormone induces ovulation? Yeah, LH. So in fact, you can also do HCG because it's actually more potent, okay? And it works just like LH. Okay, so now we have a bunch of eggs. We look at the quality of the eggs. You put them in a dish and stuff like that. Okay, it's time for the men to contribute, right? Okay, so man is there. What hormone are we going to inject the man with to get the sperm? What do you think? Stop overthinking things. Testosterone? Okay, for the men that are in here, if you ever go to an IVF clinic and they're going to ask you, okay, I need your sperm, I'm going to inject you with testosterone, or inject you with anything for that matter, just run away. That's a very bad clinic. Okay? If you give a man a room and maybe some nice music and a magazine, he will be able to provide that sperm sample under normal condition if everything is fine. Okay? So then we put sperm and egg together. We're going to watch this on the microscope, and we can look for pronuclei, polar bodies, things like that. And then we have a blastocyst, and we're going to transfer that blastocyst. How many are we going to transfer? And before you answer this, recognize that one thing, an average IVF cycle is how much money? How expensive is this? $20? Oh, yeah, no. Maybe in other countries, but let's be honest, this is the United States of America, and we're all capitalists, and we're all going to make money. And with $20, that's nothing. 15, 20,000. I mean, it really kind of depends, but that's kind of the ballpark that you need to thinking of, okay? That's one cycle. The other thing that to recognize is maybe on average, okay, the success rate is about 30%. 30%, okay? Now let's think about this question. How many of these blastocysts, because you have multiple eggs, you're not collecting one egg, and you're definitely not doing one sperm if you do a regular IVF. So how many are we going to put back? We want to make sure that the woman is pregnant. One? Wow, that's $15,000, $20,000 with a chance of 30% that it actually works. Three or four? Here in the US, there's really not any law that prevents you from how many you want to put back. So we could put 8 back and get octuplets if you're lucky, right? So usually they put more than one. Depends on the age of the woman, really. If a woman is younger, they may put less. In the United Kingdom, I think if you're younger than 30, okay, then they only would do 1. If you're older than 30, they would do 2 or 3, okay? But it's variable, okay? What you do have is with most of the IVF procedures result in multiple pregnancies, like twins, okay? You have a higher incidence of that. Is that bad? No, of course not, okay? The couple is there for a reason, okay? ICSI is something that is actually used more often, and it stands for intracytoplasmic sperm injection. So now basically what you do is take a sperm and you inject it into the egg, okay? And then you monitor that. Yes?
Would fertility pills also increase FSH and LH?
Yes, there's certain fertility drugs that will definitely can do that too, yes. Yes?
Is this what they do at like sperm banks? You just like a sperm bank to do this method, or do they do the other method?
So in what case can you imagine a situation where you would do ICSI? Well, there's different reasons. In fact, I believe ICSI is more successful than IVF. That's one. That's why IVF clinics will do that. What is another reason where you don't have a choice?
If a man has like low sperm motility or just general fertility, it won't be able to break through the.
Yep. Yeah, basically if there's a problem with sperm, so that the sperm really is not going to fertilize the egg, okay, in vitro. So in that case, you take that sperm and you inject it. Yes?
So in IVF, the sperm isn't necessarily injected, it's just put.
Yep.
in close proximity,
Yep.
then this one is actually injected.
Yes. In this case, the sperm is injected into the egg. So, okay. So that's kind of a difference. Okay, I still have a couple of minutes left, so I can squeeze in one more thing. Cloning. Okay, just because cloning is fun. Come on. Okay, usually when people talk about cloning, we talk about reproductive cloning. Okay, reproductive cloning involves a procedure called somatic cell nuclear transfer. Basically, that means a somatic cell, you take the nucleus and you put it into an egg. That somatic cell in humans has 46 chromosomes. You put it in an egg, you add some electricity to it. It sounds like Frankenstein, but really that's what it is. But you activate that egg and it starts developing. Okay, so this is how that procedure. Okay, it's kind of the procedure shown right here, but this is for humans. We don't do this in humans. Okay, this is not happening. But this is how the first cloned sheep was produced. What is the name of the first cloned sheep? You guys know this? Sorry?
Dolly.
Dolly, yes, Dolly. Dolly the sheep, the first cloned large animal. Okay, so this procedure was used for that. Horrendously inefficient. I think the success rate was something like 0.3%. Okay, so that's not very good. So we need lots of eggs, lots of this procedure, and lots of transferring it back and see if it worked. Who is Dolly named after? Do you know that? Sorry? Dolly Parton. Why would you call a cloned sheep Dolly Parton? Sorry?
Why not?
Why, of all the names you could have picked, Dolly Parton. Scientists are really lame. They have no sense, well, odd sense of humor. Okay, what is Dolly Parton known for besides country music?
Plastic surgery.
That too? Her hair? I don't want to go to hair. There's something else. Maybe the men would have noticed that more than the women. No, no, think of a physical attribute. A physical attribute. Her boobs, okay? She had big boobs. I don't know, had or has? Has. She still is. Okay, sorry. Okay, has. Okay, big boobs. The somatic cell that was used to make Dolly, guess where it came from? Mammary glands. So there you go. That's why she was named Dolly Parton. Or not Dolly Parton, Dolly. So where's Dolly now? What do you think? So Dolly was born 1996, I think. Yeah, 1996. Well, she is dead. But you could still see her in Edinburgh in the museum there. They stuffed her up. You can, if any one of you ever goes to Scotland and goes to that museum, send me a picture. Okay? Okay, that's it. That's I can squeeze in more stuff. But I know we're 1 minute over. I have 3 example exam questions. You want to do them? Really quickly. I'm going to do them quick, okay? And I'll post them as well. So if people are online are already stopping, then it's fine. Which of the following is not involved in primary sex determination? Win4, RSB1, SRY, AMH, sex chromosomes. The key things here are not and primary sex determination. Want to see the answer? Right? AMH is involved with secondary sex determination. Okay? This is the most wordiest question. There's no such question on the exam. I just need to come up with something, okay? Spermatogenesis, what it is. Which of the following indicates the correct order of germ cell differentiation in the adult testis? So, spermatocytes, primary spermatogonia, secondary spermatogonia, spermatozoa. Spermatogonias, primary spermatocytes, spermatids, secondary spermatocytes, spermatozoa. Spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, spermatozoa. Or spermatogonia, spermatids, spermatocytes, sperm. Oh, this one got cut off. E is primary spermatocyte, secondary spermatozoa, spermatids. Which one? See? Last one. From which part of the male reproductive tract could you collect sperm that can be used to fertilize an ovulated oocyte in vitro in a dish? Testis, efferent duct, head of the epididymis, body of the epididymis, tail of the epididymis. A side note, again, men, if you ever go to a fertility clinic and they want to grab a needle and do any of these things, just run away, okay? This is not how we have to collect sperm. Which one? There you go. Good luck tomorrow.